Hello, everyone. My name is Louis Nastasi. I recently completed my PhD in entomology at Penn State, and I am extremely honored to have been selected as a recipient of the Snodgrass Memorial Research Award here with the SysEB section at ESA. Unfortunately, I'm not able to be there in person with you to accept the award, but I would like to share a little bit about my dissertation research on the gall wasps. Gall wasps are a group of parasitic Hymenoptera in the family Cynipidae in the superfamily Cynopoidea that constitute a diverse array of host specialist insects that are associated with a wide variety of plants. As the name implies, gall wasps induce galls on their host plants, which are novel structures developing from the activity of that gall inducer, and within those galls, the offspring of the inducer will feed and develop. Gall wasps are extremely interesting because they're a diverse group with over 1,500 described species, and they have an incredibly diverse set of hosts across 8 different plant families ranging from oaks, roses, and herbs. And gall wasps are a really, really interesting study organism because they have incredibly diverse and complex gall phenotypes, even relative to other more diverse gall inducers like the gall midges. We can see some examples of gall phenotypes up here on the top right of the screen. And examining how these phenotypes develop and how these insects are affecting plants allows us to investigate both basic questions like how insects manipulate plants, all the way to much more directed and structured questions like how natural selection drives the evolution of gall phenotypes. And gall wasps are also very interesting because they have transitions between many different life histories all across their phylogeny. As you might imagine, most gall wasps are gall inducers, but we also have a group of gall wasps termed inquilines that can't induce their own galls anymore and instead have to develop inside galls induced by other gall wasp species. Beyond that, we also have typical insect parasitoids, which will lay their eggs inside of galls and then feed on insects already present there. And this gives us an opportunity to investigate how life histories originate and shift in diverse specialist insect lineages. So, as you might imagine, gall wasps are a really excellent insect to study because of these and other reasons. And we would imagine that they'd be an incredible model system for exploring both broad and narrow ecological evolutionary phenomena. But unfortunately, there's a multitude of systematic barriers preventing consistent application of gall wasps to these and other research areas, such as a lack of identification keys, the production of relatively few comprehensive taxonomic revisions, the lack of a phylogeny, and the presence of many, many undescribed species all across the world. And for my dissertation, I wanted to start lessening these barriers to allow gall wasps to be more broadly applied to answer all different types of questions. So I started first by inventorying the gall wasps present in North America. I started by producing a catalog to select gall wasp taxa that recovered 170 valid species in 12 genera, providing a comprehensive data set representing the geographic distribution, host plant use, host gall use in some cases, and gall phenotype data for a tremendous diversity of gall wasp species. Using this catalog, I was then able to develop a key to the genera that were included to allow for convenient and robust identification of the majority of higher gall wasp taxa present in North America. From there, I chose to conduct some more concentrated taxonomic research on a specific gall wasp group, that being the tribe Aulicidiinae, a group of gall wasps associated entirely with herbaceous plants. I conducted 2 different scale revisions in this group, one being a small scale revision of a single unit of species, the Antistrophus rufus complex, but from there, I proceeded to revise the entire North American fauna in the tribe. And as a result of these studies, we see an extreme expansion of biodiversity and taxon richness knowledge in the group, and of course, an expansion of the capacity for further study. I was able to identify and describe 6 new genera and 25 new species in this tribe. Within that diversity is a very high number of new host plant records that were not currently known for this group. And I was also really excited to identify a possible model system in these gall wasps for understanding ecological speciation and host specialization, that being a group of gall wasps inducing galls on Silphium. And to very briefly outline this system for all of you, on this slide, we see a schematic showing the diversity of gall phenotypes and some information about the host plant organ and host species associations. As we can see on the left, there's a tremendously diverse range of gall phenotypes these insects can induce, ranging from clear swellings of the stem all the way to entirely hidden chambers within the stems and the basal leaves. And we have a tremendously diverse set of host specialists inducing these different galls across 15 different host plant species, with many still undescribed and many Silphium species remaining entirely unsurveyed. So, based on our findings showing a high degree of specialization, it seems quite appealing to use this particular unit of gall wasps to answer more directed questions about how host specialization and speciation occur. From there, I then aim to conduct some broader studies affecting the whole of gall wasps, being a UCE phylogeny for the superfamily, and then using that phylogeny to establish a preliminary revision of gall wasp families. This here is the phylogeny of Cynopoidea, which includes 367 taxa and represents all higher lineages. And this tree is derived from a 50% partition matrix showing almost 1,500 loci. And I'm excited to report that I was able to recover a relatively robust tree, stabilizing the higher topology between the higher level gall wasp groups at the family, subfamily, and tribe levels. And using this data, I was able to investigate the evolution of gall induction, inquilinism, and parasitism across the superfamily. And we are currently working on finalizing some analyses aiming to estimate the number of origins of these natural histories and overall produce a more generalized framework within which questions of gall wasp evolution can be developed. So as a result of these studies conducted during my dissertation, I've been able to lead the modernization of gall wasp systematics by producing modern taxonomic studies, producing identification aids that are essential to understanding gall wasps as study taxa, and of course, producing a large-scale, robust phylogeny for understanding the evolution of this exciting group. This work would not have been possible without the contribution of a tremendous number of individuals. I want to briefly thank our gall research team at Penn State, which includes members of my dissertation committee and my advisor, Andy Deans. I want to thank some folks at the Land Institute for assisting with a lot of the Silphium gall wasp work. And I'd like to thank my funding sources, the National Science Foundation and the Society of Systematic Biologists, for supporting my work. So I would like to once again say how honored I am to be selected as a recipient of this award. And I would like to thank you all very much for being here. Thank you.